Hey guys, hope everyone's having a great weekend. This is, let's see, we're week 5 going into week 6. Oh my goodness, I can't even believe it. We're still in the cells unit. We just spent 2 weeks on models of cells, learning about the different types, prokaryotic versus eukaryotic, and what type are we? Eukaryotic. You are eukaryotic. Um, we have our prokaryotics, which are bacteria. Then we learned about the difference between plant and animal cells because those are 2 types of eukaryotic cells. And there are some very important differences between those. As we go into week 6, we're getting ready to go deeper. We're going to learn how the organelles, which are those tiny organs inside of eukaryotic cells, how they work together to help the cells stay alive. So we should know by now the individual functions, the individual jobs of each of the organelles, the tiny organs inside the cells. If we don't know that by now, we should review our digital notebook. It's on eClass at the navigation bar, very easy to access. Lots of good stuff there. So check it out because you will need to know it for our upcoming week. We'll start the week out with a little project, creating our analogy model. That's just to help remind us of what each of the organelles does and what organelles are within each type of cell, plant and animal. All right, then we will dive into how they work together to help the cells stay alive. Things like reproduction or obtaining and using nutrients for energy, stuff like that. So it's gonna be an interesting week. There were a couple of things I wanted to show you. So I'm going to actually stop this video and reset so that I can show you what I want to show you. The first thing is I wanted to show you about the plants and their roots and why root cells don't have chloroplast. So I'm going to show you that here in just a second. The second thing is I want to get started a little mini project that I'm going to start outside of school, and then as it gets closer to being finished, I will bring it into school to show you in person. But I wanted you to watch it along the way because it's very, very cool. I did this last year. I think you're gonna really like it. So hang on real tight. Actually, it's gonna be no time at all for you because I'm just gonna cut now and then recut to the next video. All right, be right back. All right, so I'm back and reset. What you're seeing is one of the many succulent is what they're called, plants that I have around my house. Usually sits upstairs in my bedroom window getting lots of light. But this one happens to be one that I can easily pull out of the soil. Now there's rocks on top you can see, but there's also some soil, a little bit of soil underneath. So this is a plant, it is a live plant, okay? We're actually trying to save these. We got these from a family member, and we're trying to, I guess, make them bigger and stronger and healthier. So this is a new project we've been working on. But you'll notice the top of the succulent plant is green, okay? This is a plant that needs light. And it actually needs not that much water. We water them once a week. That's something that's different about succulent plants than a lot of other plants. So you can see the green on top. And if you remember, plants are made of plant cells, which are eukaryotic cells. Now, the difference, well, some of the differences between plant cells and animal cells, which humans are made of animal cells because we are animals. But some of the differences are that plant cells have a cell wall, and that's so they have some more protection, and they can be upright instead of just a pile of mush because they don't have skeletons, right? Talked about that. They also have a large vacuole. And I just told you that this succulent only needs to be watered once a week. Again, that's not how all plants are, but this particular plant only needs water once a week. Now, I'm assuming the cells in this plant probably have very large vacuoles because they only need to be watered once a week. That water is being stored in the cells within the vacuole, so they can use it to keep themselves healthy as, you know, Monday, Tuesday, Wednesday, Thursday, Friday, Saturday go by, and then we water them on Sunday. And then the cells take back in the new water. All right. The other difference was that plant cells have something called chloroplast. But not all parts of the plant, because remember, there's lots of different types of cells in each organism that are multicellular. We talked about humans. We said, I know I told you guys about the brain cell, right? It looked really weird. That's because of the job it does. It has to send messages, so it has all these dendrites sticking off of it that help it to send those signals to other brain cells. And that looks very different than, let's say, skin cells that are flat, or red blood cells that look like disc. So how the cell looks in the organism, the multicellular organism, depends on its job, its function. So the structure, how it's built, depends on the function, its job. So in our plants, they have different types of cells, different types of plant cells. Now, the cells on top, where you see green, the leaves that are green are made up of cells just like every other part of it, but those cells most likely have a lot of chloroplast because there's a chemical inside of chloroplast called chlorophyll that helps give plants their green color. So any part of the plant that's green, the cells within that part of the plant probably have a lot of chloroplast. Chloroplasts are also responsible, in addition for giving the plant its green color, they're responsible for photosynthesis. This is the process where the chloroplasts take in sunlight and they add in a little bit of carbon dioxide, and then they produce glucose, which is the food for the plant. Now, chloroplasts just produce the food. In addition to the glucose, they do have some byproducts that come off, and that one of those is oxygen, which is important because the chloroplast then sends the oxygen and the glucose over to the mitochondria. And remember, the mighty, mighty mitochondria. Picture Miss Scott flexing right now because that's what I made you guys do the other day. And I did it myself too, so, you know, I'm not a hypocrite. Mighty, mighty mitochondria takes that glucose and oxygen and turns it into ATP energy. All right? So the chloroplast, which has chlorophyll inside of it, which makes the plant green, is responsible for photosynthesis. This is the process where the chloroplast take in sunlight, add in carbon dioxide, and they produce glucose, which is basically sugar. This sugar and another byproduct made through photosynthesis, oxygen, are sent over through the cell to the mitochondria, where the mitochondria performs something called cellular respiration, and that's where they turn it into energy, ATP. The cell needs energy to do anything and everything. So this is a very important process. And I'm already explaining to you how two of the organelles are working together to help make sure that the plant and the cell stays alive, or the cell and the plant stays alive. So the chloroplast, they make the food, the glucose, and then the mitochondria turns that glucose into energy. That energy is then used by all other parts of the cell to perform their jobs, including the chloroplast. So chloroplasts need energy to do the photosynthesis job. So, pretty cool, right? Kind of like a soccer team working together, or any sports team that works together. All right, now, what I want to tell you about is down here. This is the root of the plant, of the succulent. And you can see a little tinge of green here and there, but mostly it's like white or brown. Now, I told you that the cells that have the chloroplast, and inside the chloroplast, the chlorophyll, that's what makes it green. But down here, we don't really see so much green. So is it possible that we have plant cells that don't have any chloroplast at all? Yes, it is. And those are typically your root cells. So if you get a question like that, remember, the plant's not just leaves that are green or stems that are green. There's also roots underneath the soil. And the roots, you're like, how does it survive without the chloroplast to make food, to be turned into energy? Well, they're busy soaking up water and nutrients from the soil, so they have a different job. So they probably also look different than the cells you would find up here in the leaves. And part of that difference is they do not have chloroplast, whereas the plant cells up here in the leaves would have probably many, many chloroplasts. All right, so that's the first thing I wanted to share with you. I hope that made sense. I plan on bringing this little guy to work with me, and hopefully we can make him grow and at the very least stay alive together. So you guys will help to be in charge of that, and we can figure out a name for him when we get together. All right, stay tuned for part 2, which is even more exciting. It's an excellent project that I think you're going to be very excited about. It's called The Naked Egg. Get ready. All right, next up, I'm going to get us started on our naked egg experiment. I know it sounds a little weird, but I did it last year when we were talking about osmosis and diffusion, and that is coming up relatively soon. But what I did learn from last year is that this process of preparing our eggs to be, quotes, naked, can take a while. It actually took over a week. So I wanted to go ahead and get started with that just to make sure, even if they do get ready early, I can always just show it to you ahead of time. But I wanted to make sure that they had the opportunity to be fully ready for you to see what you need to see when you need to see it. I know that's a lot of very vague words, but you're going to understand very soon. Okay, so consider this part 1 of this video, and as the egg changes, I will be sending out updated videos just for you to see how it's coming along. Sorry, my words aren't working correctly today. Now, this is a very easy experiment you can do at home. I wanted to show you things you could use. You will need, of course, eggs, and you don't have to use a whole dozen. I'm just going to do that One, I want to make sure that I have enough that come out okay. And two, I also want to share this with the other teachers. So I wanted to make sure I could send at least one fully transformed egg to each other team so they could show their students. All right, so if you have 1 or 2 eggs at home, I would maybe try at least 2, just to be sure, you know, nothing happens to one of them. So if your parents will allow you to use 2 eggs for this egg experiment, right? That'd be awesome. It's great to try. Another thing you need is distilled white vinegar. A lot of people already have that at their house, and if you don't, and your parents would like to let you try this at home, it is very cheap. I think I got this at Kroger. This is Kroger brand. I bought it today. This huge thing of it was like $3. So it's very, very cheap. I actually bought 2 of these because I am doing 12 eggs. And as I said, last year, it took a little over a week to just prep the eggs. And you're gonna learn more about that in just a second. So I did actually end up using about 1.5 of these for 12 eggs. Now this year it could go differently. I could use 1 or less and it'd be totally fine because I am actually separating the eggs this year. Whereas last year I put them all in one big container. So separating them may make it take less materials. We shall see. All right, so the eggs and the white vinegar are actually what we start with, okay? You'll need some kind of container with a lid because vinegar smells really, really bad. So the container you pick needs to be large enough for the eggs and have a lid, but you also want to make sure that you can fully cover the eggs with the vinegar and that they're not floating up out of the vinegar, excuse me. Now I put a couple of other things out here as well. Corn syrup. This will be used after we get our eggs prepped. So after our eggs, basically we're dissolving the shell off of the egg, and the egg is still going to be intact. We're just getting rid of the hard part of it, but you'll see the egg will still be intact. And that's what the eggs and the vinegar and a container with a lid so you don't have to smell it are for. And you will, once you set them up in the container, you pour the vinegar over it, you put the lid on, you make sure the egg is fully covered, then you put it in the refrigerator, okay? And you check it every day. So tomorrow, in 24 hours, I will take the eggs out and I'll look at them, and if they look like it's working pretty good and the vinegar looks to be still being effective, I'll probably put them in with the same vinegar for another 24 hours, so 48 hours in the refrigerator. After 48 hours, 2 days in the refrigerator, if they're not totally ready, then I'm going to gently, carefully pour out the vinegar that's in the container and put fresh vinegar in there. And that should restart basically the chemical reaction that's occurring and help it dissolve faster. And then I'll kind of repeat the process. I'll go another day and I'll check it, and if they're still not ready, but the vinegar still looks good, I'll wait another 24 hours. And if after another 48 hours, 2 days, so now we're at 4 days, they're still not ready, I'll give it one last go with replacing the vinegar. And that's what it took me last time. But again, I had all 12 eggs, possibly more eggs than that. I might have actually done 2 dozen in one large container, and it took a long time and several times of me trading out the vinegar for it to work. But once I got a naked egg, okay, so the egg's still intact, it kind of looked like a rubber ball, a yellow rubber ball. Then I wanted to put it into a container again, so you could use the same one, just rinse it out. and then put the egg in there and then pour corn syrup over it. And you want to make sure that the corn syrup is completely covering the egg, because the egg is going to want to float. So you may even actually have to put some kind of device in there to hold it down. But we'll talk more about that when we get to that step. I just wanted to let you know that the corn syrup is not going to be used today. It will come later though. All right. After the corn syrup step, there's a step where we use water and food coloring. So if you have food coloring at home, I know a lot of people already do, and you're gonna do this at home, awesome. If not, it's pretty cheap too, $2 to $3 at Kroger. And I got the neon kind because that's the only kind I can find, but I actually already have a lot of other colors as well here at the house from all my slime making activities with Ghost Stem. So who knows what kind of colored eggs we shall be making when we get to that point. All right, so step one, basically the jars or a container, the eggs, however many you're doing, and the vinegar. That'll take a few days, possibly a week. All right, then you'll have 24 hours with the corn syrup step, so that's another day, and then 24 hours with the water and food coloring step, so that's another day. So again, it's like a week and a half to 2 weeks to do this entire project. And every step of the way, guys, I will be filming every day. I will do a short video to show you what's happening with the eggs. And once I get them to the initial step where they're naked, I'm gonna bring them in and show them to you. And I'll probably bring in the corn syrup and pour it in into the jars at the school so we can monitor them at the school at that point. The vinegar is just really, really smelly, and last year trying to leave them in the staff refrigerator, it was a mess. And I definitely don't want them in my tiny refrigerator in my classroom smelling that kind of way. So, all right. I'm gonna go ahead and pause and prep some of these jars and then return when those are ready to show you what they look like. So I just realized that I forgot a couple of important things. Number one, why is Miss Scott dissolving the eggshell off of the egg? Well, and this is very interesting. You know how most cells are microscopic, meaning we can't see them with our naked eye. Eggs are actually one of the biggest cells. Unfertilized, eggs that is. So if this egg was fertilized, meaning that what are the male ones? A rooster had come by and fertilized the female, and then the egg had a baby chicken inside of it, a chick, right? Then that would be different. But the unfertilized eggs are actually just large cells. So I want to get rid of this hard shell on the outside in order to show you the cell membrane of the cell. Now you're not going to be able to see all of the organelles inside. It's a little bit different with eggs like this. It's hard to explain in middle school terms. There's a lot of research on it. But you're not gonna see, a mitochondria and all that stuff. You're basically just going to see the cell membrane in this experiment. And then at the very end of it, once we're done going through all the processes that we are trying to show you with it, how things can move in and out through the cell membrane, which is basically what we're trying to show you. We want to first expose the cell membrane, make the egg naked. Then we will show you how things have moved into the cell. That's with the vinegar. Next, we show you how things can move out of the cell. And then finally, we show you how they can move back into the cell. And that's all part of maintaining homeostasis, right? Balance. All right, at the very end, we do, kind of destroy them because they burst sort of like water balloons. And at that point, we have colored them because they've absorbed our food coloring and water into them, and they're really fat like a water balloon. So that's kind of exciting at the very end. But the whole process is really cool. But I just wanted to make sure I pointed out to you that the whole reason we're doing this is, one, because unfertilized chicken eggs that most of us eat are cells. By dissolving the eggshell, we are exposing the cell membrane of that cell. And two, we want to show you how things can move into the cell and out of the cell through that cell membrane. That's the main points of this activity. And it's just really cool too, if I haven't said that already. It's an excellent activity. All right, one last thing, as I was prepping these, you can see I have just an egg in each jar so far. See if I can show you. So this is a very fragile egg, and I'm sorry for the noise. I'm in my garage working. But this is just a regular egg, so it's not hard-boiled or anything. If I drop it, it's going to break. So when I'm putting it into a glass container, because these are glass, so I don't know what you're gonna use, but please be careful. I turn them sideways, the glass sideways, and then, let's see if I can see this. Kind of push the egg into it down there and make sure the egg was at the bottom before tipping it upright to make sure they don't break before we even get started. All right, so I'm gonna pause you guys again to fill these with vinegar. All right, so just to show you, that took about half, maybe a little less than half of the container of distilled vinegar. So definitely if you are going to have to, if you're trying to do a full dozen, right, 12 eggs, then you will at least need one full container, because you're definitely going to have to pour it off after 2 days, I would think, and pour some new vinegar on. If you're only doing a couple of eggs, then that one jar should be more than enough, or one container. And you can already see inside of the glasses that there are bubbles starting to form around the eggs. All right, that's how the shell is going to be dissolved. So when I pull these out of the refrigerator tomorrow after 24 hours, so it's 4:30 right now, I'm gonna check on them at 4:30. If the bubbles have stopped, but there is still a lot of shell left on the egg. And I need to go ahead and change out my vinegar, because that means the reaction has stopped and the vinegar is no longer any good. If there's still bubbles going, but I still don't have a naked egg, meaning I still have some shell left, I can leave that same vinegar for another 24 hours. I'm hoping that putting them all in separate containers will make this process go faster, since all of these were in one container last year, and I can show you a picture of that too. I might post it with this video. It took a long time. I think they were competing over who got the bubbles, you know, your typical egg competition. I'm just kidding. All right, so all these bubbles going, I'm gonna go ahead and put the lids back on, and then I'm gonna place them into the carton, because that's just a nice way to carry them. And then I'm going to put that in my refrigerator and wait until 4:30 tomorrow to see what we have going on. Yeah, and I'll definitely be checking back in with you guys tomorrow at 4:30 to show you what they look like. All right, can't wait to talk to you then.